Welcome back, everybody. Thanks for rejoining us. Our next presentation is from Nathalie Munoz and Lisa Bramer on experimental design. Looks like Nathalie's got her slide up, so take it away.
Great, thanks, Scott. I'm Nathalie Munoz. I'm a scientist at EMSL, and I'm going to be talking today about selection of data based on the experimental goals. So I'll start by saying that, as John, Christine, and Scott explained, we have plenty of options to analyze samples from an omics standpoint. Some of the different options are presented in this graph, and Christine mentioned this in detail and highlighted the different days that we will focus on the different approaches. But yes, so on day 2, we're going to be talking about proteomics. And on day 3, we're going to be talking about metabolomics. And in the ABF, these are, and in the ABF and in this school, these are the two techniques or the two topics that we'll cover in lots of details. However, it's important to note that we don't need to do all of the omics with all of the samples that we get. So there are different approaches that we can take depending on the question that we want to answer. So the choice of the experiment, it's very much based on the questions that are being asked. We can use targeted approaches or we can use untargeted approaches. We can use targeted approaches when the questions are very specific. So this was the case in this study on the left with the bacteria Pseudomonas putida, in which the question was how much of a transporter, specifically a muconate transporter, MUC, was present in engineered strains. And this transporter was inserted into 3 different insertion site loci. Using targeted proteomics, we were able to compare the MUC expression levels among strains with this transporter expressed. The Y error bars represent the standard deviation of the 3 replicate samples and is plotted with respect to the growth rate that is shown. I'm not showing, that is shown here. And it also, the growth rate was measured as optical density at 600 nanometers. To targeted proteomics, synthetic heavy isotope labeled peptides corresponding to this MUC protein have to be used, and we will expand on this in tomorrow's proteomics lesson. And a targeted approach has also been used in metabolomics. So when we want to know how much of the carbon source, in this case glucose, has been consumed, we can use a targeted approach, and we just need to measure the glucose that is left in the medium after a certain amount of time. And if we want to measure how much of the product, that in this case for the set that we'll focus on is 3-hydroxypropionic acid, we have after certain days, we can use a targeted approach. So in this figure on the right, we can see the strains that we'll study in depth in the school and a time course of glucose concentration from day 0 up to day 8, and the same for the bioproduct 3-hydroxypropionic acid. This is a short detour just showing you that to assess microbial bioproduction, we can use different expressions or different measurements. So titer is the TRY that John explained before, are some of the most common metrics for when we're talking about bioproduction. Titer is the primary benchmark characterizing upstream manufacturing efficiency. It is the amount that we measure as weight of an expressed agent relative to the volume of the total upstream produced liquid that contains the agent. And if we're growing our culture of the microorganism in a bioreactor, the volume is that we use for this amount. And the data that we get is provided in terms of grams per liter. The other important metric is yield, which is the amount or the weight of the product produced per unit of the amount of substrate. So the units that we're going to use to measure yield are grams of product per grams of substrate. And usually the substrate is glucose, but we will see that we can use other carbons, other sources as our substrate. And in rate, finally, we take into account the time and we measure it as grams of product per liter per hour. So see that all of these three measurements we can use, or we can get if we're using a targeted approach. However, sometimes we have broader questions. And using multi-omics approaches on strains that have been engineered provides an in-depth look at the proteins and metabolites that are involved in both the growth and the production of target, but also not target bioproducts. So if we think about it, in the most simplified scheme of engineering the strains for 3HP bioproduction, these are the steps that we get to get to this product, to this 3HP. If we want to see it with more detail, we can change to the scheme in which we have the actual enzymes that are doing this production of 3HP and where are they coming from. But of course, we know this picture is not complete, and all of these metabolites are substrate and products of other reactions. So the same for the cis-aconitide deletion, which all of the samples that we'll be studying have. The accumulation of this cis-aconitide impacts other biochemical pathways like the TCA cycle here. And if we take into account even the bigger picture of metabolism, we can appreciate that these untargeted measurements on biased approaches using multi-omics can give a detailed look at the proteins and metabolites involved in the production of the target and non-target bioproducts. That besides aconitide accumulation that we know because of the cis-aconitide decarboxylase deletion, besides the 3HP production, and the higher expression of the proteins directly involved in these pathways, there will be other changes, alterations, repercussions in the metabolome and in the proteome of the samples as results of the modifications that we're doing. In the diagram on the right, we can see the complexity of the metabolic network. And this is in fact for Aspergillus gyrus. Oh, okay. So some of the questions that we can answer if we have an untargeted approach is if multiple events, multiple transformation events with the 3HP pathway expression constructs were reflected in an increased protein levels and the 3HP levels, if these are correlated. Other questions that we can answer is what other metabolites besides 3HP are significantly produced, and because this would indicate that the carbon is being diverted towards other pathways. How is 3HP being degraded, and also if transport is an issue. So we collected all of these different omics data sets, targeted proteomics, discovery proteomics, and the same for metabolomics, intra and extracellular, to try to get a clear picture of what is going on inside and outside of the organism. We have these broad type of questions because generally we're not changing one parameter, but we have multiple genetic modifications. We're using other carbon sources, as John explained. It can be glucose, but we can also have xylose, galactose, and even DMR or complex medium can be the carbon source. We can have, because we're growing these cells for some of the cases in bioreactors, we can control the pH versus not control. We have different aeration conditions, and we can use completely different media in the culture. We can change the amount of phosphate, use a different nitrogen source. So we want to get a complete omics study to explain the changes that are taking place. So when we follow untargeted approaches, what we do is that we report all of the proteins and all of the metabolites that are detected in the samples. These are long lists that look like this. So on the one in pink, it's going to be proteomics, how a proteomics result looks. Wait, I'm losing my cursor. Okay, how the proteomics results look, and it's just a long list of protein with the different peak areas for the proteins detected. And for metabolomics, it's something similar. We just get the identification of the metabolites. We know this takes a long process, and we will explain both of these process to get to the proteomics data and identified metabolomics data. But at the end, we just get all of these identifications, and in metabolomics, we even get unknown for the features that don't have a good match for the library. We have to process the chromatograms, which requires alignment, picking, identification, meaning it's complex and requires an effort in data analysis. And also that we need to have something that we need to have clear is that these are relative levels. These are not absolute quantifications, and we can compare levels of the same protein or of the same metabolite between samples. And we will go in depth about this in days 2 and 3. The untargeted workflow is in general is global in scope and outputs data that's related to a comprehensive cellular metabolism. Another omics that we use routinely at BNNL, but we won't have a full day dedicated to, is lipidomics. So I want to spend some short time discussing lipidomics. And in lipidomics, it's usually done also in the untargeted global mode. And in this case, peak apex intensities of lipids detected in the samples from vastly different subclasses are reported. So this is what we're observing in the table. For the fungi Aspergillus, we haven't done a lot of lipidomic studies, but again, as we were saying, the omics that you use depends on the question that you have. But for other microorganisms like Rhodosporidium torulioides, which is an oleaginous carotenoic yeast, which has been studied as a model organism for lipid-based and terpene bioproducts, we have used untargeted lipidomics approaches, and this is the key tool to evaluate engineered Rhodosporidium torulioides. As with the other omics, there is considerable efforts from the bioinformatics side, and we needed to process the data. We're currently using for this analysis pipeline MCMine for chromatographic alignment and gap filling. For identification, we're using liquid, and liquid, that's how the liquid graphical user interface looks. This liquid program utilizes a library of more than 35,000 lipid molecular species that comprise 70 different subclasses. It is customizable, so it allows for the addition of novel lipids to the ones that are already in the library. It has the ability to identify coeluting lipid species, which happens a lot with these types of samples, and it typically identifies between 400 and 500 lipids. Other techniques that we can use are stable isotope assisted metabolomics. In this one, we use heavy isotope labeled tracers like 13C, 15A, 15N, 18O to support the studies of biochemical mechanisms. And by measuring the incorporation of the label, in this case, it's glucose labeled in 6 carbons or in carbon 1, we can, by measuring the incorporation of the label into the metabolites that are produced downstream of the tracer, we can identify or discern pathways that are involved in biochemical processes, metabolite reaction rates, and in the case of synthetic biology, it's extremely helpful because we can characterize non-model hosts, we can identify wasteful byproduct pathways, and we can also identify pathway bottlenecks. So this is exactly what we need to continue the DBTL cycle. So as final notes, we have to remember that there are a large number of analytical platforms and omics that are available to study synthetic biology. That the choice of the experiment is very much based on the question that it's being asked. So we cannot think that because we have a sample, we want to try all of the omics, but sometimes the targeted approach is what we need. And when we're assessing bioproduction, sometimes those are initial numbers that we can get. And then for our next cycle, we can use the other omics that we didn't do in the first one. We can also say that targeted and untargeted approaches provide valuable information in synthetic biology study each of them has its angle, and it's very helpful for the type of cases and problems that we're studying. But integrated multiomics can provide possible solutions to low production efficiency by identifying metabolic bottlenecks or pathway sinks, and can also contribute to understand how these engineered biological systems function, because there are many of them that are not non-model organisms. And finally, that the data processing and analysis efforts are needed in all of the omics. And we're going to learn about this in day number 2 and number 3. And also the struggle of synthesizing the data and integrating it and deriving precise actionable items is known. And we will learn about this on Thursday and Friday. However, an aspect that is usually disregarded or overlooked is the experimental design. And if we want to get useful and accurate answers, we must think about this. We have to think clearly and design the experiment in a correct way before embarking into the adventure of trying all of these different omics. So this is what Lisa will discuss in the next presentation. Thank you.
Seeing a few questions pop up on Discord. Natalie, I don't know if you're on Discord. You want me to read them out?
Sure, or I'll go to the channel and I can reply there. I don't, or yeah, maybe we can answer after both of our presentations.
All right.
Okay, I think I've successfully unmuted myself.
Thanks, Lisa.
Yeah. Yeah, thanks, Natalie. That's a great introduction to thinking about experimental design. So my name is Lisa Bramer. I'm a statistician, data scientist here at PNL. So I want to talk a little bit today about often the forgotten aspect of when we decide to run experiments, which is thinking about how we're going to design those experiments themselves. And the general steps that we tend to go through is, so first, as experts in your field, you choose the biological system that you're interested in, right? And you're going to balance some key items there in terms of physiological relevance, reproducibility, and complexity of the system you're thinking about. And then just as Natalie was talking about, then what you want to do in order to answer questions and hypotheses that you have about that system is to select what type of data should be generated in order to answer those questions or test those hypotheses. And when we think about the hypotheses that we're going to be tested, we can think about what sorts of treatments are we going to apply. Are we going to run different time point data at different time points so that we can observe a process throughout time or at a static point in time? And what happens rarely, but I want to sort of suggest, is that it's often super useful to run preliminary experiments, or at least use preliminary data to inform your experimental design setup. And the attributes of your response in these preliminary experiments might help you select kind of appropriate time points where something of interest is happening when we don't know enough about that system a priori, or for example, relevant doses that we should be using. I've definitely, as a statistician, had people come to me and bring data that they want to test for differences in doses, but those doses are either not high enough or all too high to actually see any differences. And the final part of this that I think we all come back to is determining the number of replicates that we should be running per sort of treatment or time point. I just want to check, Leanne, I see that there's something happening in the chat. Am I okay to just ignore?
Yeah, I think you can keep going and we'll answer in real time.
Okay. So I first want to kind of answer the question of why am I kind of harping on this idea of replicates? So replication is one of the core things that you need to establish any sort of statistical significance in any analysis that you do. And by having enough replicates, we can guard against the loss of the significance because of things that you might not be able to account for, like sample-to-sample variation, or something may go wrong in the experimental technical realm of things. And I know that as a statistician, we also get a bad rap of saying we just want all the data possible. I will say that anyone that you're working with should also realize that there is a balance between cost and the quality of the data that you get. More is not always better, but if you don't have it enough, the data won't be able to be used in a useful way, and your reduced price is not going to be worth the effort at all. So we need to find that right balance. And what I'd like to mention up front is that there are different types of replicates that capture different sources of variability. So often you'll hear people talk about technical or injection replicates, and those are samples that come from the same physical sample or specimen, but might get run multiple times on an instrument. And in general, many of the types of data that we generate in EMSL, like proteomics and metabolomics, the technology and the instrumentation is good enough and at a sophisticated enough point that really doing technical replicates in a lot of these cases doesn't allow you to gain a whole lot. There's not a ton of variability from just running the same sample through the instrument, but it is something to keep in mind if you're using a newer type of instrumentation, or something that hasn't been established for a long time, like some of the proteomics and metabolomics instruments we have. And the other type of variability that we think about is biological variability, and that comes from when we collect samples from different people or different leaves or different soil cores or whatever it may be. We expect that there's going to be some inherent variability there. So whenever we're talking about experimental design, the main thing that a grant or a research proposal or any sort of researcher typically uses to think about how many replicates do I need is they will use statistical power as a quantity to calculate and help determine how many replicates they should run. So I'm going to maybe make my case one more time. Why do we bother thinking about all of this, right? Well, we want to find scientifically meaningful effects. And the number of replicates is not just about finding significance, but how big of an effect size is needed to find significance. And it's really a consideration of money. So if you undersize your study, you're wasting resources because you're not going to get useful results. But if you oversize your study, right, you've wasted money as well. And there are ethical issues that are connected if you're using humans or animals. And maybe the most convincing argument that I can give you is that grant reviewers are going to be looking for this when you propose and say, please give us money to study this system. This will be one of the key questions that they ask. So I want to take a little bit of time and talk about the key components that go into determining statistical power. So to get us all on the same page, we have to think about some terms. So if we're talking about hypothesis testing or tests of significance, right, a hypothesis is just some claim or belief that we want to test. And in science, we have two competing hypotheses, a null hypothesis, which is what we assume to be true inherently, and the alternative hypothesis, which is usually what we want to show, or the significance relationship that we want to find if there is evidence. And when we think about hypotheses, the null hypothesis is sort of what you think of if someone is on trial for a criminal case. We assume that they're innocent until proven guilty. You have to have enough evidence in whatever's happened to say, I'm no longer going to assume this, and instead I'm going to conclude that this person is guilty. So science works the exact same way. We're going to assume that there are no differences or whatever our null hypothesis might be, unless the data provides us with more evidence to say, oh, we do see differences, and we're no longer going to assume that there aren't any. I'm just going to, throughout these slides, use the notation of H sub O and H sub A to denote the null and alternative hypotheses. And it's usually stated in terms of some population parameter. So that parameter, probably the most common thing you'll know of would be like a mean for a certain group. It may be a proportion or another quantity like that, but we have different types of hypotheses where we're testing if things are equal, not equal, or greater than or less than. So the last sort of piece that we need all to be on the same page is that there are different types of errors that we can make when we do hypothesis testing. So if we are going to reject the null hypothesis in favor of the alternative, that does not guarantee that the alternative is actually true, even if you have very strong evidence. So along the left here, we have the truth, and along the top, we have the decision that you might make based on your data. The two types of errors we have are, let's suppose that your null hypothesis is true, but if we reject that null hypothesis, that's called a type 1 error, denoted by alpha. Some people will refer to this as a false positive. And the other error we can make is if the alternative hypothesis is actually true, but we don't reject the null hypothesis, that is a type 2 error, or what some people refer to as a false negative. And these two errors are sort of rooted in what we think about when we're doing power calculations. So by construction, hypothesis testing is sort of denoted and meant to limit the rate of false positives or type 1 error rates. We use this significance level alpha, right? Most of you know that the magic threshold that we use for alpha is 0.05, because hundreds of years ago, someone decided that 5% was an acceptable error rate. And the key thing here is that we choose what that significance level is ahead of time. And we might be doing multiple tests. So if you're doing multiple tests, that error rate for one hypothesis test does not necessarily hold if you're doing a bunch of them. But in general, the more tests that we do, the more likely we're going to be to find a false positive. That type 2 error rate that I talked about, which is if your alternative is true, but you don't reject the null, that is a function of your sample size, the effect size, and highlighted here the significance level. So that significance level is back to your type 1 error rate. So the key here is there's a trade-off between type 1 and type 2 error rates. So if you wanted to be really stringent and never make a mistake, that's going to come at, and set alpha to 0.001, that's going to come at the cost of never finding it, or very rarely finding anything that's significant, and likely making more type 2 errors. So really this replication issue and the types of errors we can make, we're sort of brokering between the two things to find the optimal balance. So all of that background is to say, what is statistical power? So statistical power is 1 minus that type 2 error rate. And it simply is just the probability of rejecting your null hypothesis when the alternative hypothesis is true. So making the correct decision of finding something that's significant. And the thing I really, if you get nothing else out of this talk, the thing that you need to get out of this is that your statistical power is first and foremost rooted in how you decide to design your experiment and the tests that you're going to conduct. So you need to know those things a priori. And the other factors that play into this is that type 1 error rate, the effect size you would be able to want to find, how much variability is in your data, and how many samples or replicates you have, right? Those all interplay with your statistical power. So if we're going to calculate power, there's some things we can kind of cross off the list easier than others. So, you know, we talked about type 1 error rate and that there's this trade-off between type 1 and 2 errors. Typically, the lower you make your type 1 error rate, the lower the power you're going to have if you hold everything else constant. Just based in literature and sort of the scientific community, typical values that we use for type 1 error, 0.05 or 0.1. It can be domain dependent, but I would say in, you know, biology and omics data, 0.05 is pretty common. And effect size and variability sort of go together. So the effect size is how big of a difference, let's say you're testing for a difference in 2 means for your favorite protein. How big of a difference needs to be there in order for you to be able to say there's a significant difference. And the variability is just how much variability is inherently in your observations. So if you're evaluating a difference in means, that measure of variability is the variance of values from your same group or your same treatment. And it's really this ratio of effect size and variability that determines the power of your experiment. So if you have data that is inherently more variable, that's going to mean your study will be less power. And if you want to be able to find really, really small effect sizes, that's also going to mean you have less power. So what are sort of the typical values of these? Well, it again sort of determined by data and or domain knowledge, but I will say that oftentimes with the types of proteomics and metabolomics that are generated at EMSL, often like a 2-fold change in means is a typical sort of expected difference that one might see for some organisms. And variability really, unless you have a very well studied and understood system, this is where it comes back to being really important that you have some sort of data to be able to calculate variability from, whether that's, you know, a preliminary experiment or a past experiment. And finally, then the 2 other things that tend to go together are sample size and power. So these are usually the things that we want to estimate. We typically are asking the question, if I have X number of samples, what is my power? Or how many samples do I need for 80% power? That 80% threshold seems to be something that is fairly accepted in the community, particularly for things like NIH grants. The one thing I want to point out that's a key difference here is in most domains, we have one estimate of variability. So you're only studying one variable, so you get one power calculation. But in biology, when you're generating omics data, you almost always have multiple response variables, so multiple proteins or metabolites, and you get multiple power calculations to make life even more complicated. So just a quick example, let's suppose that I've got some salmon that are being smoked, and I want to measure the mean level of benzopyrene. And I want to test that level of benzopyrene is at least that of if we smoke the salmon in a shed. You know, in this experiment, we smoke samples of N salmon and a teepee, we compare that to a shed. We might ask the question, how many replicates do I need in order to get 80% power, and perhaps a twofold change is reasonable. These are the types of questions we're asking when we're calculating statistical power. And I just really briefly want to mention, in a case like this, it can be beneficial to talk to a statistician, where you could actually increase your statistical power if you have a fish and it's possible to smoke part of the same fish in each of your two treatments. You actually gain more power that way by having paired samples. So just an example of what power calculations might look like for omics data. Again, if you have many measurements, many proteins, metabolites, you get many estimates of variability from your data, and thus you get many power calculations. So for example, this previous data set, we have 200 metabolites. We can do 200 power calculations because each of those metabolites has a different variance or standard deviation. And the results of statistical power are typically calculated as how many samples you have per treatment group in order to reach some level of accepted level of power, like 80%. I will say this is something that if you need to actually think about power and how many replicates you need, the key resources you have, the first suggestion I have to you is to contact your favorite statistician if you have someone at your disposal. But this is especially true if you're dealing with things like time course experiments, sequencing data, data that might have missing values, or imbalanced groups in your different treatment groups. If you're sort of in a very typical kind of experimental design, I've listed some online resources for you here, GPower and this powerandsamplesize.com. They both have calculators that will allow you to input those key quantities that I've mentioned in this talk, and will allow you to see sort of how well your power is or how many replicates you need, depending on the inputs. And if you're familiar with R programming, there are a couple of very common packages in the stats and the PWR package that you can use. And to tie this all up, I just want to give you a few other considerations. So you might ask, what if my sample sizes are already determined? So we know that that can be true. You may have what you have, or you may have a fixed amount of money, and your samples are what they are. In that case, if you're writing a grant, typically what you do is you can calculate the power based on expected effect sizes, and that again could come from preliminary data, or perhaps a publication that you or the scientific community has put out and shown what types of effect sizes are seen for a certain type of data. And so that question is, what power will I have to detect a twofold change in mean expression given my sample size? Or you can take your sample sizes and the power that you need to achieve and calculate how big or small of an effect size can be detected based on that required power. And all of these calculators that I had on this previous page can do these calculations as well. A second question you might have is, what if I don't have preliminary experiment data? What I will tell you is that power can be calculated for some hypothesis tests under sort of a worst case scenario. For example, if your outcome that you're measuring are proportions, we know that the highest level of variability will be if the proportion you observe is 0.5. You can do a power calculation under that scenario. My best suggestion to you otherwise is to identify variability values from literature, or see if you can find data from another study where they're generating the same type of data, but perhaps the samples aren't the exact same types of samples you're using, that will at least give you a better idea than just sort of doing nothing at all. The other things you should think about and talk to folks about is, are you going to have missing data? So for example, proteomics data, it's quite common to have samples and values that go missing for various reasons, either below the limit of detection or other issues. All power calculations assume that you have no missing data. I mentioned, are you testing more than one biomolecule? It goes the same way. Are you testing more than 2 groups? You might need to consider multiple test corrections. And if you're doing hypotheses that aren't dealing with straightforward things like means and proportions, let's say you're doing trend analyses over time, variability is really not a straightforward calculation, and having example data is key. And the last sort of tip I will give you is that if you're contemplating, should I add another treatment group or should I add more replicates because you're balancing a budget? In terms of statistical power, the answer is always that you should do more replicates within a group rather than adding another group. All you'll be doing in that case is spreading your statistical power even thinner. With that, I will wrap up and see if there are questions.
The questions will be in the chat or in Discord.
I don't know if you're looking at Discord, Lisa. There's a recent question if alpha and beta values are related.
They are not inherently related to one another. So they are only related in that when you try to control for one, that affects your ability to control for the other. So you can make, all the false positives and false negatives that you want, and they're not necessarily inherently related by your hypothesis testing. But if you're looking to ensure that you can achieve or you stay below one of these 2 error rates, it's going to come at the cost of the other one increasing inherently. Essentially, a type 1 error rate makes it more and more difficult to say, I have found something significant. And if it's harder and harder to find something significant, then that means it will be harder and harder to reject your null hypothesis. So your type 2 error rate will likely increase because you won't be correctly rejecting the null hypothesis in a lot of cases. I know statistics is everyone's favorite topic, so.
There's a fair number of questions about metabolomics that I see Nathalie addressing in Discord.
Yes, I'm checking that one. Youngmo will check it too.
I see a question about EMSL ficus resources, if they're available to generate labeled substrates for stable isotope tracing. I think the substrates generally are purchased, and that's usually done by the user and we get the labeled samples sent to us for analysis. That's generally the path taken.
Yes, that's correct.
Yes, that's generally the way it works. They're cultured in other labs by our collaborators, and they send us either the pellet or the supernatant that we can analyze here.
I would like to answer Lee Yuan-Hu's question to the metabolomics. When you design the experiment, will you use more refined media, avoiding using rich media such as yeast extract? We do both, actually, but at the very beginning, we want to understand their mutation, metabolic engineering better. It is much better to use a single carbon source with a minimal media. That is helpful to understand how their intracellular metabolism changed. However, in reality, there's some collaboration. They have to add some additional carbon or nutrients like a yeast extract to improve their production efficiency or increase the final product amount. In that case, we are tracing their metabolism changes as well as tracing the medium samples over time course. So for example, if we add a yeast extract at the beginning, we also trace what components of the yeast extract were consumed or some other byproducts secreted out to the medium. So we will cover this topic on Wednesday session, but we trace both the intracellular metabolism and extracellular metabolism as well. So we will cover all the metabolites to get the final view of metabolism of the host.
I do see a question from Aaron Simmons here about outlier tests and removal in omics data. I think it's a great question, and I'll give you the hopefully satisfying answer that if you come to the Thursday session where Kelly Stratton and I are going to talk about actual data processing for the omics data, we will talk about formal tests that you can do for outliers and when it is subjectively and or objectively okay to remove them. But it's a great question. And when we talk about power, I mentioned you can calculate variability across 200 metabolites. We typically don't believe that we should have to achieve that the standard deviation for all of those metabolites are informative, as we know some of them are just not, haven't maybe potentially gone right. So we will take, a percentile, something like the 75th percentile, and say, if we can get 80% power for 75% of the metabolites in this preliminary study, that we should have good coverage. But we will talk about both biomolecule and sample outliers on Thursday.
So I don't know if you guys have seen it in the chat, but some of the earlier questions are how sensitive are the isotope detection approaches for lipids and the approximate percent label of substrate needed? Low or more than protein CYP or high like DNA CYP? And is the process of labeling metabolites error-free? If not, how do we overcome that?
Yes, I didn't understand the error question one. For the lipidomics, I have to say I have not done, we have usually done labeling of polar metabolites, but not for the lipids. So I would have to check with the lipid side, with the lipid experts, and I'll get back to you and reply in the Discord.
I do see a question that says, can the addition of time series samples compensate for the lack of biological replicates to increase power? So my answer there is that if you have enough time points, you can basically switch from doing sort of a traditional ANOVA type comparison of means, where you might actually be able to fit some sort of regression or time course model. And those types of models do have inherently better power than just sort of traditional ANOVA. So it can be a nice trick to yeah, try to maximize your data by doing it over a span of time series and decreasing replicates if you've set it up correctly. And what I would just suggest is that you talk to a statistician, or some of those online tools do have some regression power calculation if you're sort of savvy enough as well.
Lisa, in that case, are you assuming a steady state or pseudo steady state during the time course sampling? Because obviously, I mean, biology is varying as from growth to production to, senescence and things like that.
Yeah, I mean, if your process is, going to be approximately linear, or even, it doesn't have to be linear, it could be, say a quadratic, but something that isn't going to have a drastic, if you've got a drastic jump, you have to have enough time points covered in that jump time period. So there is some nuance there about sort of, the mechanism and how quickly things are changing, and make sure you're capturing that. But that's a good point, John. But in general,
you can think of an example, right? When we're in high production phase, that lasts for quite a large number of hours. So you could sample across that quite intensively and achieve that objective, I think.
Right. Yeah. So it is, this would be a case where hopefully you've got some preliminary data or some published data to give you an idea of what's happening in a time course, so that you know where to allocate your time points. But yeah. So Kim asked, what could be better multiple testing option to apply in metabolomics and proteomics? So we will talk a little bit about this on Thursday as well. But when you're doing multiple testing, we tend to, especially if you're in the discovery phase, we tend to do a multiple testing correction within a biomolecule. And if you're measuring, a couple hundred of things in a true discovery phase, we often will not apply a multiple testing correction in that direction. But so the answer will vary depending on which direction of correction that you want to do, and we will cover that on Thursday. Yeah, and then there's a question about, do you have a rule of thumb for the minimum number of sample replicates for standard discovery omics experiments? Most of the field defaults to n equals 3. Yeah, Aaron, I appreciate that question. So n equals 3 feels like you know better than to do n equals 2, but you didn't try that hard. No, I know that there can be constraints on that. There really isn't a general rule of thumb, because it is truly dependent on the system that you're studying. But the best sort of global rule that I can give you is that if you're generating proteomics data, where you can get quite a bit of missingness, especially in the shotgun approach, then having 5 replicates per group really safeguards you because of that missingness. In the case of other types of data where you have less missingness, like metabolomics and lipidomics that are generated by typical sort of GC and LC, 3 generally will work, but it again is really dependent on that variability with the actual organism and media type. So, we know things like if you've got cell lines are going to have way less variability than if you're analyzing human plasma. So.
Did you see the question on Zoom, Lisa, about looking at spatial gradients?
How about spatial gradients for regression analysis rather than time series? Spatial gradients are, I guess I feel like I might need to know a little bit more, but yeah, about the type of analysis that you're planning on doing. So, you know, spatial gradients have a similar flavor to time series, but if you're working in 2 dimensions instead of 1, then you're going to require more samples inherently than time series. So time series is sort of an analog to spatial statistics. Soil depth trends, gotcha, thank you. Yeah, if you've got enough samples there to do a regression analysis, that's going to maximize your power. The problem with this, right, is to do regression, that means you need, you know, at a very minimum 5 sort of different depths or time points where you're really going to be able to start capturing any sort of spatial or time series trends. You might not need as many replicates at each of those depths or time points, but you know, if you only do 2 time points or 2 spatial sort of locations, you know, you get perfect regression, obviously, and it doesn't tell you anything. So, but it's pretty analogous to time series in terms of the actual power, to answer your question, Ricardo.
All right, I think questions are kind of slowing down, and we're coming up on a few minutes away from noon our time, and that is scheduled to be our extended break, lunchtime for us here on the Pacific time zone. And the afternoon is devoted to breakouts with the students we accepted into the tutorial sessions for the afternoon. So this morning, this is the round out, the close down of our open sessions for the broad community. I really appreciate all the questions, because it shows that this sort of preface to what you're going to hear later about proteomics and metabolomics and types of data and how you analyze it and how you get it into a model is things you guys are excited to hear about already. You're asking questions that, you know, we're going to start to touch on lots more topics throughout the week. So excited to see that you guys are interested in learning more. Anything to add, Scott, before we completely close out?
Nope. Thanks everybody for tuning in today. We look forward to more sessions tomorrow. It's been a lot of fun. I hope you've enjoyed it as much as I have. And thanks to all of our speakers today.
Yes, thank you, everybody. Hopefully we'll see you all back tomorrow.
Yes.